Okay, question 5. The endocrine system is also involved in responding to changes in the external environment. For example, adrenaline is released in the fight or flight response or the stress response. When adrenaline arrives at a liver cell membrane, it binds to a receptor. Inside the cell, a cascade of events leads to glycogenolysis. Explain how this response would be of use to a rabbit that has seen a predator. Now, this is one of those questions where I bet some of you have written some really clever stuff, but it doesn't actually answer the question. So it's already said to us, adrenaline is released. The adrenaline arrives at the liver cell membrane, binds to a receptor. Once inside, a cascade of events leads to glycogenolysis. Now, all of that causes that, which causes that, which causes that, which causes that. Explain how this response would be of use to a rabbit that's seen a predator. So all we're talking about, the response, we're talking about how is glycogenolysis useful to a rabbit that has seen a predator. They just like to make it wordy and confusing because they're mean. Okay, so how is glycogenolysis useful? So it's only 3 marks. So first bullet point, glycogenolysis is the splitting of glycogen into glucose. So you've got to practice your G words if you couldn't remember that, okay? Lysis means splitting, and then we've got glycogen. So it's splitting that stored glycogen in the liver into glucose, okay? Next, explain how this response would be of use, okay? So there's 1 mark. The glucose will be released into the blood. And will travel around the body. Again, back to the question, how would it be of use to a rabbit that's seen a predator? So it's already said about fight or flight response. So glycogenolysis is splitting that stored glycogen into glucose. That glucose is then in the blood, okay, which is important. What's it then going to do? It's going to go to all the different effectors that are going to be involved in the stress response. So I'm just going to go with skeletal muscle. You could say it's going to go to lots of different areas where it's going to cause effects. So we know about the different effects of the stress response. So pupils dilate so that they can see better. What else? Heart rate will increase, breathing rate will increase, and so on. I'm just going to go for skeletal muscle because I think it's the simplest answer. So the glucose can then be used by skeletal muscle in respiration to release energy. Again, I've not fully answered the question yet. How is this of use to the rabbit? How is releasing energy of use to the rabbit that's seen a predator? To release energy, which can then be used for muscle contraction to run away. Okay, next question. All 3 types of muscle in the rabbit's body contribute to the fight or flight response. Complete the table below by filling in the blank boxes. Organ: heart, leg muscle, arterial to the liver, type of muscle, action of muscle in fight or flight response. So in the heart, what are the three types of muscle? We've got skeletal, which is the same as conscious or voluntary muscle. We've got smooth muscle, which is the same as unconscious or involuntary muscle. And we've got cardiac muscle. So heart, that's definitely cardiac. Leg muscle will be skeletal. Action of the muscle. So all it's asking for here is, well, I suppose it's not really action of muscle. Okay, so leg muscle, skeletal. It's so that the action of the muscle will be to contract to allow movement. So run away, for example. Arterial to the liver, that's going to be contract. Hold on, let me think. No, to the liver, we want it to relax, don't we? Relax, to increase diameter, to increase blood flow to liver, Why would we want that to happen? So that we've got more glucose getting to the liver, because that whole process of glycogenolysis, that requires glucose itself to split the glycogen down. So we've got to get more blood to the liver to start that process and get more being released. Okay, question II. Muscles in the rabbit's leg are made up of units. The figure is a diagram of one unit. So we've got our A band, or heavy band it's sometimes called. We've got our I band, which is sometimes called our light band. And then we've got our H zone, which is myosin only. A band is the whole of the myosin, and where the actin overlaps as well. Name the protein. Oh, what was the question? The thick lines in band A represent a protein. So this is myosin, and the light ones are actin filaments. So the question asks you to name A. So the answer is A. Was it A? Thick lines in band A. Yes, so myosin. The contraction and relaxation of human muscles relies on interactions between different proteins within muscle cells. In skeletal muscles, the proteins are arranged in bundles known as myofibrils. The figure below shows simplified diagrams of some of the proteins present in muscle myofibrils. So this is muscle contraction, which Miss Fleming went over with you. And I uploaded my lessons as well onto Teams, so you can have a look at those, and a silly little role play that I came up with, which you'll have to imagine because of Corona. Okay, using the letters A, B or C, identify the following proteins on the figure above: tropomyosin and troponin. Tropomyosin is the rope-like one. It kind of has rope in there. Tropomyosin, ropomyosin. Oh, but I suppose it's in that one as well. It's a longer word for a longer molecule. Tropomyosin is the long one. Okay, so that's B. And troponin, or troponin, however you want to say it, is the little one, which exposes its binding sites so that it changes the shape of the whole myofibril. So troponin is C, and that's a shorter word, isn't it? So it's the smaller molecule. Okay, last one for this video. The image below is a transmission electron micrograph of a section of skeletal muscle. Which row A to D shows the correct labels? So again, like with the brain question, I would work it out before I look and get myself confused. So I'm going to turn it and I'm going to have a look. So here is one sarcomere, which means that this is my M line, M for middle. Then we've got our Z's either side, okay, which shows the end of one, the end of the next. Then we have got our, we will have our myosin will be just down the middle there. M for middle and also for myosin. And then we'll have our actin, which will be here. What else does it want you to label? And then this thing here, well, it's got a label here for glycogen granules. So I would suggest that these are mitochondria because there's loads of them. There's not just like one or two big ones. So you know it's not going to be a nucleus, okay? And we know that there will be loads of mitochondria in skeletal muscle cells. Mitochondrion, okay? So organelle T, we're going with mitochondrion. So that one is likely. We could have made a mistake though, so we're going to keep looking through. Region U, where's that? Here, so this dark band. We have said that we think that that's the ends of one sarcomere, so our Z line. So we think it's probably that one. Not our light area, not our heavy area. So it's looking good. Region V, have I written over it? Where's V? Where is V? I must have written over it. Oh, it's there. Region V, so yeah, so that's one myofibril, isn't it? With lots of those actin and myosin filaments. We've got loads of them in a bundle. So definitely that, not that, not that. And finally, the region between X and Y, so this whole thing here, that's going to be a sarcomere. So that fits quite nicely, I think. So B is the answer to that one. Hopefully you got that because none of the words are sort of repeated. So I think it's quite clear that there's only one answer. So hopefully you got that one. The answer is B. Okay, I'm going to stop the video there.